Hello everyone, welcome to my channel Priyanshi Biology Basics, and in this video we will understand chapter number 5 of class 9 science, which is the fundamental unit of life. So while examining a thin slice of cork, Robert Hooke is a renowned scientist who in 1665 examined a slice of cork. Cork, here we are talking about bark of a tree. So he examined the bark of an oak tree and saw that the structure of cork resembled the structure of a honeycomb. Honeycomb means मधुमक्खी का जो छत्ता होता है. So he saw that just like structure of honeycomb, cork also consists of many little compartments, means many little areas, small space, small box-like structures are there. Many box-like structures are there. And he examined this through his own self-designed microscope. And Robert Hooke called these boxes, these little compartments, those small spaces, those areas as cells. And cell in Latin means a little room. That's why he named them cells. And this may seem to be a very small and insignificant incident, but it was important in history of science because it was first time that it was noticed that an organism is made up of separate units joined together. Now, what are living organisms made up of? To understand this, there is activity 5.1 in which we need to prepare the temporary mount of onion peel, and then we need to observe it under compound microscope. So we will see this kind of structure. We can see that these small compartments are the cells, and inside the dot-like structure is the nucleus, which we will study about the nucleus in the further chapter. So here it is now said that if we take onion of two different sizes, like हम एक छोटा अनियन भी ले और बड़ा भी ले और दोनों का टेम्पररी माउंट हम प्रिपेयर करें, तो क्या जो साइज में, शेप में, विल देयर बी एनी डिफरेंस इन कि छोटे अनियन के जो सेल्स हैं या फिर और जो बड़े अनियन के जो सेल्स हैं उन दोनों के सेल की शेप में, साइज में कुछ डिफरेंस होगा? सो द आंसर इज दैट द सेल्स विल ऑल लुक द सेम regardless of the size they come from, and these small structure were termed as cells. And also there are single cells that live on their own, means there are two type of organisms, unicellular and multicellular. Unicellular organisms are the organisms which are made up of single cell only, and multicellular organisms are the organisms which are composed of multiple cells. So there are single cells also that live on their own, and with the invention of magnifying lenses, it led to the discovery of the microscopic world and was known that single cell may constitute the whole organism, like amoeba, Chlamydomonas, Paramecium, bacteria, and there is one more example which is yeast. It is the exception case of fungi, and it is also a unicellular organism. So in multicellular organisms, many cells group together in a single body and perform different functions in different body parts, and those are called as multicellular organisms. And the examples are fungi, plants, and animals. So generally, cells, like many cells all together, form tissue. The tissue form organ, all the organs form organ system, which ultimately forms an organism. A multicellular organism. Now to understand next part, we need to first understand the cell theory. So this was presented by Shirlin and Schwann. So they said that all animals or plants are composed of cells. And second, that cell is the basic unit of life. Further, Rudolf Virchow, he proposed the idea of omnis cellula e cellula. Means he did, like, his idea led to further modifications in the cell theory. So he proposed the idea of omnis cellula e cellula, which means that all cells arise from the pre-existing cells. मतलब जो नए सेल्स बन रहे हैं वो पुराने सेल्स को ही डिवाइड करके बन रहे हैं. लेट्स टेक एन एग्जांपल ऑफ zygote. So fertilization of egg with sperm will form the zygote. Now the zygote which is formed is the single celled, and that single celled zygote will form the multicellular organism. How? Like, it will divide in various planes, horizontally, vertically, many times. It will divide in various planes and then will form the multicellular organism. So by this we can say that all the cells do arise from pre-existing cells. Now we can understand this, that every multicellular organism has come from a single cell. How? Because cells divide to produce cells of their own kind, and all cells thus come from pre-existing cells. Now in activity 5.2, so in activity 5.2 we have to prepare various temporary mounts of different parts of plant. Leaf peels, tip of roots of onion and peels of onion. And we need to see whether the shape and size of the cell in different plant parts vary. Like, the size is same, the shape is same, or is it different? So we will see that the shape and size of different plant part varies because they all are specialized to perform different function. Like leaf pills, they are specialized for the food preparation purposes. Roots are specialized for the absorption purposes. So उनके shape और size में there will be difference. So before we move on to next, let's understand this more to know part. So in this, first it is written कि in 1665 Robert Hooke discovered the cells. Then in the 1674 Leeuwenhoek, here we are talking about scientist Anton von Leeuwenhoek. So he discovered free living cells in pond water. So actually he saw very tiny structures. He saw very tiny structures in the pond water and he gave them the term animalcules. Why? Because those structures seems like tiny organisms to him because of their appearance and movement. Then in 1831, Robert Brown discovered the nucleus. In 1839, Purkinje gave the term protoplasm. Protoplasm is actually the fluid substance of the cell, which includes the cytoplasm, the fluid substance, and inside the cytoplasm there is presence of nucleus and some other cell organelles. So it includes all these. There is one more term which is protoplast. Protoplast is a cell without cell wall. Means it will not include cell wall but will include cytoplasm, nucleus, cell organelles as well as cell membrane, which is plasma membrane. Then it has been explained about the cell theory. And then it is said that with the discovery of electron microscope in 1940, it was able to understand complex structure of the cell. Cell itself has a very complex structure and the organelles present inside the cell are they have very intricate structures. So with the help of electron microscope, it was able to understand each and every component of those organelles as well. Now, cell organisms can also have cells of different kind. As we said, different parts of the plant have different cells. Their shape and size varied. Like, we can take the example of human body as well. In the figure it is given smooth muscle cell, blood cell, nerve cell, bone cell, sperm, fat cell, ovum. So we can say कि ये जो cells हैं वो body के different parts में present है. Nervous system है वो nerve cells के through work कर रहा है. ब्लड सर्कुलेटरी सिस्टम है वो ब्लड सेल्स के थ्रू कर रहा है. दे ऑल हैव द डिफरेंट पर्पस इन द बॉडी. So the shape and the size of the cells are related to the specific function they perform. Like some cells also change shapes. For example, let's take the example of amoeba. The amoeba is a unicellular organism and is formed of single cell, and that shape of the cell usually changes. In some cases, the cell shape can be fixed as well, like in case of nerve cells. Now, each living cell has the capacity to perform certain basic functions. And how does a living cell perform all these basic functions? Because there are components present in the cell. Cell is composed of various components. For example, it has plasma membrane, it has nucleus, it has cell organelles, and there are different cell organelles, and each cell organelles perform different functions through which a living cell performs various functions. For example, in human body, if we talk about, heart is there to pump blood, stomach is there to digest food, so each have their different function, and the specific components are there in the cell which is responsible to perform these functions. These are called cell organelles. Some cell organelles are there to form the new material, some are there to clear up the waste material, and they perform various functions. Now, what is a cell made up of? Now, if we study the cell under a microscope, we will generally come across three main features, which are plasma membrane, nucleus, and cytoplasm. And all the activities, the cytoplasm also includes a cell organelles are also present inside. So all these activities which occurs inside the cells and interaction of the cell with its environment, with the outside environment, or within itself also, due to these components only it is possible for them to interact. So let's understand about first component, which is plasma membrane. It is also called as cell membrane and is also called as plasma lemma. So this is the outermost covering of the cell. Generally in animal cell it is the outermost covering, but in plant cell the outermost covering is of the cell wall. This plasma membrane allows or permits the entry and exit of some material in and out of the cell. अब ये outermost layer है तो यही entry और exit को regulate करेगी, and only of some material. That's why we call it selectively permeable membrane, because it allows the entry and exit of only selected material. Like it will only allow useful materials to go inside the cell, and will allow harmful or waste secretions which are present inside the cell to move outside. Now, how this movement takes place? Generally, there are two transport mechanisms through which the movement takes place across the plasma membrane. The first is active transport and second is passive transport. So active transport is a rapid process. Why? Because in this case energy is required, means ATP is required, and the movement is against the concentration gradient, means from low to high concentration. And this movement against the concentration gradient can only be regulated if energy is present. And there are the example like glucose, amino acids, or ions like sodium and potassium ions can only move across the plasma membrane through this active transport mechanism. Now through this passive transport mechanism, now it is a slow process because there is no energy required, and movement is down the concentration gradient from high to low. That's why it is a slow process and does not require energy. And some gaseous molecules like oxygen, carbon dioxide, and some water molecules can move through this process. So generally there are two methods through which the passive transport can take place, that is diffusion and osmosis. So first let's understand about the diffusion process. Generally, molecules like carbon dioxide and oxygen can move across a cell membrane by the process called diffusion. And there is movement from region of high concentration to region of low concentration in this case. So here we can take the example of diffusion. Key substances like CO2, it is a cellular waste and requires to be excreted out of the cell. So CO2 gets accumulate inside the cell, and in outside environment the concentration of CO2 is low, and inside the cell the concentration of CO2 is high. So according to the movement from high to low concentration, the CO2 gas will move from inside the cell to the outside environment. And same case will happen with oxygen as well. In case of oxygen, the internal environment of the cell has low concentration of oxygen, and the outside environment of the cell have high concentration of oxygen. That's why there will be the movement of oxygen from high to low concentration, means oxygen will move from external environment to the internal environment of the cell. And that's why diffusion plays an important role in gaseous exchange between the cell as well as the cell and its external environment. Now water also obeys the laws of diffusion. So the movement of water molecule through selectively permeable membrane from high concentration to low concentration is osmosis. The movement of water across the plasma membrane is also affected by the amount of substance dissolved. Movement of water is also affected by the presence of solute in them. Thus, osmosis is the passage of water from region of high concentration to region of low concentration through selectively permeable membrane till the equilibrium is reached. Till on both the sides the amount becomes equal, the osmosis will take place. Till the amount of solvent molecule becomes equal, the osmosis will take place. Now what will happen if we put an animal cell or a plant cell in a solution of sugar or salt in water? So to understand this, let's understand about three types of solution: hypotonic, hypertonic and isotonic solution. So in hypotonic solution, we can see this is a beaker and inside there is solvent molecule. These are the solvent water molecules. These green are the solute particles, and inside there is a cell. हाइपोटोनिक सॉल्यूशन के केस में जो आउटसाइड सॉल्यूशन है, सेल के बाहर का जो सॉल्यूशन है, उसमें वाटर की कॉन्सेंट्रेशन ज़्यादा है और सॉल्यूट की कॉन्सेंट्रेशन कम है, और सेल के अंदर वाटर की कॉन्सेंट्रेशन कम है और सोल्यूट की कॉन्सेंट्रेशन ज़्यादा है. ना अकॉर्डिंग टू द रूल हाई टू लो concentration movement of water, there will be the movement of solvent molecules from outside solution to the inside of the cell. बाहर से सॉल्यूशन अंदर जाएगा बिकॉज वाटर की कॉन्सेंट्रेशन बाहर ज़्यादा है और सेल के अंदर कम है, जिसकी वजह से सेल विल स्वेल. नाउ इन हाइपोटोनिक सॉल्यूशन, हाइपोटोनिक सॉल्यूशन के केस में जो सॉल्यूशन है उसमें सॉल्यूट की कॉन्सेंट्रेशन ज़्यादा है और वाटर की कॉन्सेंट्रेशन कम है, और सेल के अंदर के एनवायरनमेंट में सॉल्यूट की कॉन्सेंट्रेशन कम है और वाटर और जो सॉल्वेंट मॉलिक्यूल्स हैं उनकी कॉन्सेंट्रेशन ज़्यादा है. तो देयर विल बी मूवमेंट फ्रॉम इनसाइड द सेल टू द आउटसाइड सॉल्यूशन. देयर विल बी मूव फ्रॉम इनसाइड द सेल टू आउटसाइड द सॉल्यूशन. उस केस में जो सेल है वो श्रिंक हो जाएगा. नाउ इन आइसोटोनिक सॉल्यूशन, आइसोटोनिक सॉल्यूशन के केस में सेल के बाहर भी और सेल के अंदर भी जो वाटर का कॉन्सेंट्रेशन है वो सेम रहेगा और सोल्यूट का भी कॉन्सेंट्रेशन सेम है. देयर विल बी द वाटर मूवमेंट जो है वो सेल के अंदर से सेल के बाहर के एनवायरनमेंट में ऐसे बैक एंड फोर्थ मूवमेंट विल बी देयर, बट देयर विल बी नो नेट मूवमेंट बिकॉज इक्विलिब्रियम ऑलरेडी अचीव हो गया इस केस में. Equilibrium has already been achieved in this case. Now, so here it is also written, first is hypotonic solution. If the medium surrounding the cell is high water concentration, means that the outside solution is dilute. So in that case, cell will gain water by the osmosis. Water molecules are free to pass across the cell membrane in both the direction, but more water will come into the cell than will leave. सेल के अंदर से बाहर वाटर और बाहर से अंदर सेल के मूवमेंट होगा, बट सबसे ज़्यादा मूवमेंट होगा जो बाहर का सॉल्यूशन है, वहाँ से जो मॉलिक्यूल्स की मूवमेंट वो ज़्यादा होगी कंपेयर टू जो सेल से बाहर के एनवायरनमेंट में हो रही है उसके. अब इस केस में जो होता है हाइपोटोनिक केस में सेल स्वेल्स अप. सेकेंड कम्स आइसोटोनिक सॉल्यूशन. सो आइसोटोनिक में हमने देखा कि concentration is exactly the same on both the sides, so there will be no net movement. So in this case cell will stay of the same size. Now if the medium has, now there is hypotonic solution. Hypotonic में medium has low concentration of water, so it means कि जो outside solution है वो बहुत concentrated है. So in this case cell will lose the water and ultimately cell will shrink in this case. So thus osmosis is a special case of diffusion. Osmosis itself is the case of diffusion because उसमें high to low concentration movement हो रही है, no energy required. But the difference is कि water molecules का movement है, solvent molecules का जो movement है उसे हम osmosis कहते हैं. और generally diffusion में gaseous molecules का भी movement होता है. But osmosis is particularly for the movement of water molecules. नाउ इन एक्टिविटी 5.3. सो एक्टिविटी 5.3 में एक्चुअली क्या दे रखा है कि हमें एक एग लेना है, डी शेल्ड एग, कि हमें एग का शेल निकाल देना है और सबसे पहले हम उसे प्योर वाटर में रखेंगे. अब प्योर वाटर में सॉल्यूट की कॉन्सेंट्रेशन 0 है. अब सॉल्यूट की कॉन्सेंट्रेशन 0 है मतलब वाटर का कॉन्सेंट्रेशन हाई है प्योर वाटर में, बिकॉज जहाँ पे सॉल्यूट का कॉन्सेंट्रेशन ज़्यादा होगा वहाँ पे वाटर का कॉन्सेंट्रेशन लो होगा, जहाँ पे सॉल्यूट का कॉन्सेंट्रेशन कम होगा वहाँ पे वाटर का कॉन्सेंट्रेशन ज़्यादा होगा. तो प्योर वाटर में वाटर का कॉन्सेंट्रेशन ज़्यादा है और इनसाइड द एग वाटर का कॉन्सेंट्रेशन कम है बिकॉज सॉल्यूट पार्टिकल्स भी हैं. तो उस केस में हाइपोटोनिक वाला केस है, तो दिस एग विल स्वेल. सेकेंड केस में हमने उसको सॉल्ट सॉल्यूशन में रख दिया, मतलब अब हाइपोटोनिक सॉल्यूशन का केस हो गया है. तो उस केस में एग विल श्रिंक. In activity 5.4, the same activity has been done with the raisins and apricots. इसमें भी जो result होगा वो same होगा. So here are given the examples of osmosis. Unicellular freshwater organisms and most plant cells tend to gain water through osmosis. So unicellular freshwater organisms also gain water through osmosis, and plant cells by absorption of water by plant roots. Through roots they absorb water, and these two are the example of osmosis. Thus diffusion is important in exchange of gases as well as water in the life of a cell. In addition to this, cell also obtain nutrition from the environment, and these nutritious molecules move in and out of the cell through a type of transport which requires energy. Here we are talking about the active transport mechanism. Now they are given some more points about the plasma membrane, that it is flexible and is made up of organic molecules called lipids and proteins. and we can observe this structure through electron microscope. Now this flexibility of the cell membrane, plasma membrane, also enable the cell to engulf the food inside it from the external environment, and this process is called as endocytosis. And one of the example of the organism which perform this endocytosis process is amoeba. Now the next component we are going to study is cell wall. Plant cells, in addition to the plasma membrane, have another rigid outer covering called the cell wall. The plant cells are the cells which have cell wall on their outer side. Apart from plant, fungi as well as bacteria also have cell wall, but animal cells do not have cell wall. The outermost covering in animal cell is plasma membrane. So the cell wall lies outside the plasma membrane and composed mainly of cellulose. दूसरे components भी होते हैं, for example hemicellulose, but जो mainly जो majority amount जो होती है cell wall में वो होती है cellulose की. It is a cellulose. It is a complex substance which provides structural strength to the plants. Now when a living plant cell, so here's kind of activity, like when living plant cell loses the water through osmosis, there is shrinkage or contraction of the content of the cell away from the cell wall. Osmosis में जो सेल वाटर लूज करता है तो सेल श्रिंक हो जाता है. जो प्रोटोप्लास्ट है वो श्रिंक होके एक साइड पे मूव कर जाता है. स्पेसिफिकली जब बाहर का जो मीडियम है वो हाइपरटोनिक हो और अंदर का जो सेल का जो मीडियम है वो हाइपोटोनिक सिचुएशन में हो, तो इस फिनोमिना को जब ऑस्मोसिस के थ्रू जब सेल से वाटर लूज कर जाता है और सेल श्रिंक हो जाता है, कॉन्ट्रैक्ट कर जाता है, तो उस फिनोमिना को वी कॉल इट इज प्लाज्मोलाइसिस. एंड ये हम एक एक्टिविटी के थ्रू देख सकते हैं. तो इस एक्टिविटी में हमें एक रियो लीफ लेनी है. तो फर्स्ट व्हाट वी नीड टू डू इज़ वी नीड टू प्रिपेयर अ माउंट ऑफ द रियो लीफ, और उसके जो हम सॉल्यूशन प्रोवाइड करेंगे उसे एक कंसनट्रेटेड शुगर या सॉल्ट सॉल्यूशन प्रोवाइड करेंगे. मतलब हम एक हाइपरटोनिक सिचुएशन क्रिएट कर रहे हैं अराउंड द सेल, एंड देन वी विल ऑब्जर्व द माउंट अंडर द माइक्रोस्कोप. ठीक है. इन सेकेंड इसमें क्या करना है, फर्स्ट वी नीड टू बॉयल द रियो लीव्स एंड देन वी नीड टू प्रिपेयर द अमाउंट, and then same as before we are going to put the strong solution of the sugar or salt around the mounted leaf. Now what will be the outcome? तो हम देखेंगे कि जो लिविंग सेल्स के अंदर हम ऑब्जर्व करेंगे प्लाज्मोलिसिस, बट डेड सेल्स में we will not observe plasmolysis because dead cells में जो plasma membrane होती है it does not remain functional, जिसकी वजह से osmosis होगा ही नहीं. That's why dead cells में no change will be visible, but living cells में we will see कि जो प्रोटोप्लास्ट है सेल का that will move to one side and will get shrink, will get contract. Cell walls permit the cells of plants, fungi and bacteria to withstand very dilute hypotonic external media without bursting. मतलब अगर आप किसी भी सेल को अगर हाइपोटोनिक सॉल्यूशन में रखोगे, हाइपोटोनिक मतलब कि उसका जो वाटर पोटेंशियल है वो सेल के वाटर पोटेंशियल से ज़्यादा है, तो उस केस में क्या होगा, जो वाटर है वो सेल के अंदर मूव करेगा बाहर के एनवायरनमेंट से. तो जब वाटर सेल के अंदर मूव करेगा तो वो वाटर सेल वॉल पे एक प्रेशर जनरेट करेगा और उस प्रेशर को हम कहते हैं टर्गर प्रेशर, और वी कैन कॉल इट एज हाइड्रोस्टैटिक प्रेशर. और उसके एक्सचेंज में, उसके रिटर्न में, काउंटर अटैक में जो सेल वॉल है वो भी एक प्रेशर जनरेट करेगा और उसको कहते हैं वॉल प्रेशर. और सेल वॉल की इसी एक्टिविटी के कारण कि वो एक इक्वल प्रेशर जनरेट करता है अगेंस्ट द स्वॉलन सेल, इसी एक्टिविटी के कारण the plant cells or the fungi or the bacteria are able to withstand much greater changes in the surrounding medium. मीन्स उनके सराउंडिंग मीडियम में कुछ अनवांटेड या कुछ डिफरेंट चेंजेस हैं तो उससे वो कॉप अप करता है. बट एनिमल सेल्स में ऐसा नहीं होता. ये प्रॉब्लम हमें देखने को मिलती है बिकॉज उसमें सेल वॉल नहीं होता. तो चांसेस होते हैं कि जो सेल है वो ज़्यादा स्वेल होने के कारण वो बर्स्ट हो सकता है. Now the next component is nucleus. So earlier in the earlier chapter, we read about the temporary mount of onion cells, right? तो उसमें हमने देखा था there was a dot-like structure, that was the nucleus. So यहाँ पे एक एक्टिविटी की गई है with the human cheek cells और उस पर आयोडीन या फिर आप कोई भी स्टेन फॉर एग्जांपल मिथाइलिन ब्लू और सैफ्रनिन यूज़ कर सकते हो. और एक वी नीड टू प्रिपेयर द अमाउंट ऑफ द ह्यूमन चीक सेल इन दिस केस. सो व्हाट वी विल ऑब्जर्व इन दिस केस कि जो न्यूक्लियस है, साइटोप्लाज्म या फिर जो डिफरेंट कॉम्पोनेंट है सेल के वो अलग अलग स्टेन होंगे. मतलब कुछ डार्कली स्टेन्ड होगा, कुछ लाइटली स्टेन्ड होगा. तो जनरली जो न्यूक्लियस पोर्शन होगा वो हमें डार्कली स्टेन्ड मिलेगा. न्यूक्लियस पोर्शन विल बी डार्कली स्टेन्ड. और एक्चुअली इसका रीज़न क्या है कि कोई पोर्शन डार्कली स्टेन्ड है, कोई लाइटली स्टेन्ड है? इट इज़ बिकॉज इट इज़ रिटन है दैट इट इज़ अकॉर्डिंग टू द chemical composition that different regions of the cells get colored differently. So let's understand about the nucleus. That it has a double layered covering called nuclear membrane. इसमें ये एक double membranous structure है. And the nuclear membrane has pores which allow the transfer of the material. और उस double layered nuclear membrane में there are pores present through which transfer of the molecules from nucleus to cytoplasm to nucleus occurs. These nucleus contain chromosomes and these chromosomes are rod shaped structure. Initially chromosomes तभी visible होते हैं when the cell is in dividing state. अगर cell dividing state में नहीं है तो what we observe is the chromatin material जो कि एक thread like structure होता है. So these chromosomes which are rod shaped structures are only visible when the cell is about to divide. Now these chromosomes contain the information, the genetic information which is transferred from parents to the next generation. And chromosome contain this information in the form of DNA molecules. So chromosomes are actually composed of DNA and protein. And DNA molecules are specifically the molecules which contain information necessary for constructing and organizing cells. Means they contain information which is required by the cell to function, to grow and to divide properly. Now these DNA contain some specific fragments which are functional and those are called as genes. These are the hereditary units. These genes are the hereditary units. So now in a cell which is not dividing, DNA is present in the form of chromatin. Just I told you that they are visible as entangled mass of thread. And when the cell is about to divide, chromatin materials gets organized into chromosomes. These chromatin material gets condensed into chromosomes. And जब cell divide हो जाता है then decondensation occurs और जो chromosomes है वो again chromatin material में convert हो जाते हैं. Now the nucleus plays central role in cellular reproduction, the process by which single cell divides and form two new cells. It also plays a crucial part along with the environment in determining the way the cell will develop and what form it will exhibit at maturity. मतलब न्यूक्लियस बहुत ही इम्पॉर्टेंट इन्फॉर्मेशन न्यूक्लियस के पास एक बहुत इम्पॉर्टेंट इन्फॉर्मेशन होती है इन सिंपल टर्म्स जो कि सेल को एक तो कंट्रोल करने में, वी कैन से दैट न्यूक्लियस इज अ कंट्रोलिंग सेंटर, लाइक सेल कैसे वर्क करेगा, कैसे फंक्शन करेगा, कैसे डिवाइड करेगा, और जैसे हम पता है कि हर एक इंडिविजुअल सेल की वर्किंग ऑल टुगेदर, सेल्स ज्वाइन करके एक ऑर्गेनिज्म फॉर्म होता है। तो दैट्स हाउ एन ऑर्गेनिज्म फंक्शंस। जो हम लाइक एक ह्यूमन बींग है, उसकी जो फंक्शन करता है, और जो भी रिफ्लेक्स सेक्शंस हो गए या फिर जो भी मूवमेंट या जो भी हो है, the main information is present inside the nucleus. In some organisms like bacteria, न्यूक्लियर रीजन ऑफ द सेल इज़ पुअरली डिफाइंड। बैक्टीरिया में न्यूक्लियर रीजन पुअरली डिफाइंड होता है, मतलब देर इज़ एन एब्सेंस ऑफ न्यूक्लियर मेम्ब्रेन। और अनडिफाइंड न्यूक्लियर रीजन को हम न्यूक्लियर कहते हैं। मतलब जो प्रोकैरियोट्स होते हैं, फॉर एग्जांपल बैक्टीरिया, उनमें न्यूक्लियस, लाइक हम तो यूकैरियोट्स में कॉल इट एज न्यूक्लियस, बट प्रोकैरियोट्स में न्यूक्लियर मेम्ब्रेन एब्सेंट होती है। तो एक अनडिफाइंड स्ट्रक्चर है, मेम्ब्रेन नहीं है, तो एक स्पेसिफिक स्ट्रक्चर नहीं है, तो उस केस में वी हैव गिवन द टर्म न्यूक्लियोइड। And such organisms are called as these are called as prokaryotes, which lack nuclear membrane. Now the one which have nuclear membrane, they are eukaryotes. So the difference between the prokaryotes and eukaryotes, ki undefined nuclear region is in prokaryotes and there it is called nucleoid, but in eukaryotes there is defined nuclear membrane, and that's why there we call it as nucleus. Now another thing, prokaryotic cells also lack other cytoplasmic organelles which is present in eukaryotic cell. So the another point कि prokaryotic cell में जो cytoplasmic organelles होती है, for example chloroplast, mitochondria, ये नहीं होती है, but eukaryotic cells have these organelles. So many of the function of such organelles are also performed by poorly organized. So कुछ दूसरे parts होते हैं, there are other structures in the prokaryotes which are very poorly organized, जो कि उनके functions perform करते हैं. Like for example chlorophyll. Now photosynthetic prokaryotic bacteria, वो बैक्टीरिया होते हैं जो कि अपना खाना खुद प्रिपेयर कर सकते हैं, तो दे रिक्वायर क्लोरोफिल, राइट. सो जो क्लोरोफिल होता है, तो उस केस में क्या होता है कि दैट इज़ एसोसिएटेड विद मेम्ब्रेनस वेसिकल. मेम्ब्रेनस वेसिकल मतलब जो प्लाज्मा मेम्ब्रेन होगी, प्लाज्मा मेम्ब्रेन की ही इन्वेजिनेशन होगी, उसकी ही इन्वेजिनेशन से एक मेम्ब्रेनस वेसिकल फॉर्म हुआ है, तो उसमें ये वर्क करता है, उसमें ये क्लोरोफिल रिलेटेड वर्क, उसमें ये खाने बनाने वाला काम होता है. देयर इज़ नो पर्टिकुलर ऑर्गेनेली, लाइक जो यूकैरियोट्स में वी हैव क्लोरोप्लास्ट इन द प्लांट सेल्स, बट इनमें नहीं होता प्रोकैरियोट्स में. ओके, लाइक यूकैरियोटिक सेल्स में प्लास्टिड्स होते हैं, हम आगे पढ़ेंगे प्लास्टिड्स के बारे में. तो उनमें प्लास्टिड्स होते हैं, क्लोरोप्लास्ट इज़ अ टाइप ऑफ प्लास्टिड. सो ये होते हैं, बट प्रोकैरियोट्स में वी डोंट हैव अ स्पेसिफिक ऑर्गेनेली. कुछ मेम्ब्रेनस वेसिकल्स होते हैं विच आर द इन्वेजिनेशन ऑफ द प्लाज्मा मेम्ब्रेन. सो हेयर इज द picture given of a prokaryotic cell, that we have cell wall, we have plasma membrane, we got ribosomes, and we got nucleoid. You can see that the structure of the nucleus, an undefined structure. Now there is cytoplasm. Now when we look at the temporary mount of onion peel or human cheek cell, we can see the large region of each cell enclosed by the cell membrane. अब हमने जब स्ट्रक्चर देखा अनियन सेल का या फिर ह्यूमन चीक सेल का, तो वी सॉ दैट सेल मेम्ब्रेन जो है दैट इज़ इंक्लोजिंग अ लार्ज रीजन, और जिसके अंदर न्यूक्लियस भी प्रेजेंट है, तो वो लार्ज रीजन इज एक्चुअली द साइटोप्लाज्म. Cytoplasm is a fluid content, and it contains many specialized cell organelles, and these organelles perform specific functions for the cell. Now cell organelles are also enclosed by the membranes, and in prokaryotes, beside the absence of nuclear region, we also don't have cell organelles present. membrane-bound cell organelles present. But in eukaryotes, we do have a nuclear membrane as well as membrane-bound organelles. So here it is given what is the significance of the membrane. So it has been explained taking virus as the example, that virus lack any membrane, right? And therefore it do not show any characteristics of the life, right? But when it enters the living body, it starts multiplying, it starts replicating, it starts using that living cell machinery to replicate and invade the area. So without membrane, virus is nothing. It does not show any characteristic, but only when it enters a living cell, then only it starts working. So here it is given the difference between prokaryotic cell and eukaryotic cell. So prokaryotic cell size is 1 micromolar, but eukaryotic cell is much larger with 5 to 100 micromolar range. In prokaryotic cell, nuclear region is not defined and called as nucleoid. But in eukaryotic cell, nuclear region is well defined and surrounded by the nuclear membrane as well. And in prokaryotic cell, there is single chromosome. But in eukaryotic cell, there are multiple chromosomes. In prokaryotic cells, membrane-bound cell organelle is absent, but in eukaryotic cell, membrane-bound cell organelle is present. In prokaryotic cell, nucleolus is absent, but eukaryotic cell nucleolus is present. So nucleolus is a structure which is present inside the nucleus. Now the next is cell organelles. Every cell has a membrane around it to keep its own content separate from the external environment. And large and complex cells, including cells from the multicellular organism, need a lot of chemical activities to support their complicated structure and function. So these multicellular organisms have very complex, intricate structure. And to support that structure and to make it function properly, to make an organism function properly, various metabolic activities takes place inside the cell. And these different metabolic activities takes place in different membrane bound little structures, which are called as cell organelles. So in this chapter we will read about the following cell organelles: endoplasmic reticulum, Golgi apparatus, lysosome, mitochondria, plastids and vacuole as well. So the first is endoplasmic reticulum. Endoplasmic reticulum is a large network of membrane-bound tubes and sheets. It looks like long tubules or round or oblong bags. The ER membrane is similar in structure to that of the plasma membrane. So let's first understand that endoplasmic reticulum, this is the structure of the endoplasmic reticulum. This is actually the nucleus, and the green colored line represents the endoplasmic reticulum. So here the endoplasmic reticulums are attached to the nucleus. So these endoplasmic reticulum can be of three types. Their structure can be of three types. First is cisternae. These are flat sheets like structure which are arranged parallel to each other, and they mostly have ribosomes on them. The second is vesicles. They are oblong, round, spherical like structures. And then there is tubules, and these tubules are the branched structures. So endoplasmic reticulum can be found in any of these structures. Now endoplasmic reticulum is actually of two types: rough endoplasmic reticulum RER and smooth endoplasmic reticulum, which is SCR. So rough endoplasmic reticulum looks rough because it has ribosomes attached to it. And the ribosomes which are present in all active cells are the sites of protein manufacture. Therefore, we can say that rough endoplasmic reticulum helps in formation of proteins. Play a crucial role in proteins formation. The manufactured proteins are then sent to the various places according to its need. Then comes SCR, smooth endoplasmic reticulum. It does not have any ribosomes attached to it. That's why its structure is not rough, and therefore it is called a smooth endoplasmic reticulum. And it plays a role in manufacture of fat molecules or lipids. Some of these proteins and lipids are used in formation of cell membrane as well. So we can say that it helps in the formation of the membrane. So this is the process of the membrane biogenesis. Like membrane formation में proteins और lipids भी help करते हैं. Some other proteins and lipid function is, and proteins और lipids के बहुत various functions होते हैं, multiple functions होते हैं. तो कुछ membrane formation में help करते हैं, कुछ enzymes form करते हैं, hormones form करते हैं. ये नहीं कि इनका एक ही फंक्शन होता है, मल्टीपल फंक्शंस होते हैं. तो SCR मेजरली फैट मॉलिक्यूल्स, लिपिड्स, इनके फॉर्मेशन में हेल्प करता है और प्रोटीन्स की फॉर्मेशन में RER हेल्प करता है. सो फर्स्ट लेट्स अंडरस्टैंड द डिफरेंस बिटवीन RER एंड SCR in detail. So RER is rough endoplasmic reticulum. It mainly consists of cisternae. So RER is majorly composed of cisternae. Other structures may be present as well, but they are majorly composed of cisternae. Smooth endoplasmic reticulum is mostly composed of tubules and vesicles only. Rough endoplasmic reticulums, in this case ribosome is attached on the cytoplasmic surface, but in SCR ribosomes are not attached. RER synthesize the proteins because ribosomes are attached, and ribosomes are the site for the protein formation. But SCR, as we saw, that they synthesize lipids and fat molecules. RER is majorly found near the nucleus. They are connected to the nuclear envelope. Just like we saw, कि this is the nucleus और endoplasmic reticulum nuclear envelope से attached है और ribosomes attached है, means these are the RER. But जो smooth endoplasmic reticulum होते हैं, they are found in the peripheral region of the cytoplasm. We can say near the plasma membrane. तो वो cytoplasmic region में मिलते हैं. So thus, one function of the ER is to serve the channels for the transport of materials between various regions of the cytoplasm or between the cytoplasm and the nucleus. The ER also function as cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. बहुत सारी various metabolic activities ER में भी होती है, जैसे हमने कहा प्रोटीन फॉर्मेशन, इसका भी एक बहुत इंट्रिकेट मैकेनिज्म है, तो वो भी ER में होता है. ट्रांसपोर्टेशन होती है एंडोप्लाज्मिक रेटिकुलम से गोल्गी कॉम्प्लेक्स में जाता है, लाइक मॉलिक्यूल्स जैसे ट्रांसपोर्ट होते हैं. इट्स अ लॉन्ग प्रोसेस. In the next is in the liver cells of the group of animals called vertebrates, SCR plays crucial role in detoxifying many poison and drugs. यहाँ पे SCR का एक और फंक्शन देखने को मिल रहा है कि जो वर्टिब्रेट्स के जो लीवर सेल्स होते हैं उसमें ये डिटॉक्सिफिकेशन में भी बहुत इम्पोर्टेंट रोल प्ले करता है. द नेक्स्ट इज गोल्गी apparatus. So Golgi apparatus was first explained by the scientist Camillo Golgi, and that's why it was named Golgi apparatus. So it consists of a system of membrane-bound vesicles arranged approximately parallel to each other in the stacks called as cisternae. तो जो Golgi apparatus होते हैं वो भी एक फ्लैटेंड सैक्स लाइक स्ट्रक्चर होते हैं. हमने अभी सिस्टर्ने का स्ट्रक्चर देखा एंडोप्लाज्मिक रेटिकुलम में. इट इज़ सिमिलर टू दैट इन द गोल्गी. फ्लैटेंड शीट्स लाइक स्ट्रक्चर होते हैं जो कि पैरेलल अरेंज्ड होते हैं. दीज मेम्ब्रेन्स ऑफन हैव कनेक्शंस विद द मेम्ब्रेन ऑफ ER, और ये भी ER से कनेक्टेड ही होते हैं समवट. जैसे मैंने कहा ना कि अगर मॉलिक्यूल्स का ट्रांसपोर्ट होना है तो जैसे ER से गोल्गी में जाता है, गोल्जाई लाइक कहते हैं ट्रांसपोर्टेशन, पैकेजिंग, मॉडिफिकेशंस का काम करता है उन मॉलिक्यूल्स के जो ईआर से उसके पास आए हैं. सो इट इज़ फाउंड नियर द ER and therefore constitute another portion of complex cellular membrane system. तो देयर इज़ अ सिस्टम कॉल्ड इज एंडोमेम्ब्रेन सिस्टम. लाइक एक कॉम्प्लेक्स होता है मेम्ब्रेन का, तो उसमें न्यूक्लियर मेम्ब्रेन, एंडोप्लाज्मिक रेटिकुलम, गोल्जाई एंड लाइजोसोम्स ये एक मेम्ब्रेन सिस्टम फॉर्म करते हैं. नाउ द मटेरियल सिंथेसाइज्ड नियर द ER ऐसे मतलब मैंने अभी बताया जैसे कि जो ER में जो मटेरियल सिंथेसाइज हुआ है वो इट इज़ पैकेज्ड and dispatched to various target inside and outside the cell through the Golgi apparatus. उसका फंक्शन क्या है गोल्जाई का? स्टोरेज, मॉडिफिकेशन एंड पैकेजिंग ऑफ द प्रोडक्ट्स इन मैनी वेसिकल्स. तो वो वेसिकल्स जो ER से उसके पास प्रोडक्ट्स मॉलिक्यूल्स जो भी आए हैं बन के, उनकी मॉडिफिकेशन, पैकेजिंग करना, उनको ट्रांसपोर्ट करना वेसिकल्स के फॉर्म में. In some cases, complex sugar may be made from the simple sugars in the Golgi apparatus. Golgi apparatus में simple sugar से complex sugar भी बनने का काम होता है. Golgi apparatus is also involved in the formation of lysosomes. ये इस वाले पैराग्राफ में Golgi के बहुत सारे functions दे रखे हैं. But we read that storage में modification of the molecules, packaging of the products, complex sugars formation, lysosomes formation. तो ये इसके मल्टीपल फंक्शंस हैं. लाइजोसोम्स. Structurally, lysosomes are membrane-bound sacs which are filled with the digestive enzymes. These enzymes are made by RER. Lysosomes are kind of waste disposal system of the cell. Lysosomes, first thing is they are made from the Golgi complex and they are membrane-bound sacs. Only single membrane is present. They are not double membrane bound organelle, they are single membrane bound organelle, and they consist of some digestive enzymes like hydrolytic enzymes which are made by RER. Lysosomes are kind of waste disposal system of the cell. They help the cell to keep it clean by digesting any foreign material as well as worn out cell organelle. कुछ भी डैमेज्ड मटेरियल है, कुछ भी डैमेज्ड ऑर्गेनल है, वो उसको क्लीन करने में, रिमूव करने में हेल्प करता है. फॉरेन मटेरियल्स एंटरिंग द सेल सच एज बैक्टीरिया और फूड एज वेल एज ऑर्गेनल्स, ओल्ड ऑर्गेनल्स एंड अप इन द लाइसोसोम्स which break complex substances into simpler substances. Lysosomes are able to do this because they contain powerful digestive enzymes capable of breaking down the organic material. तो ये इसको डाइजेस्ट कैसे करते हैं? बिकॉज इनमें डाइजेस्टिव एंजाइम्स होते हैं और वो बहुत पावरफुल होते हैं कि वो उसके कॉम्प्लेक्स जो उन ऑर्गेनल्स में, फॉरेन मटेरियल में जो कॉम्प्लेक्स सब्सटेंस होंगे वो सिंपल सब्सटेंस में ब्रेक डाउन करते हैं. और मोस्टली ये जो एंजाइम्स होते हैं ना, ये एक्चुअली हाइड्रोलिटिक एंजाइम्स होते हैं. During the disturbance in cellular metabolism, for example, when the cell gets damaged, lysosomes may burst and the enzymes digest their own cell and therefore they are called as suicidal bags. जब सेल डैमेज हो जाता है तो उस केस में लाइसोसोम्स बर्स्ट हो जाते हैं, और अगर लाइसोसोम बर्स्ट हो गए तो उसके अंदर जो एंजाइम्स हैं वो बाहर आ जाएंगे और वो फिर डाइजेस्टिव एंजाइम्स वो खुद के ही सेल को डाइजेस्ट कर लेंगे. इसलिए इसको हम सुसाइडल बैग्स भी कहते हैं, बिकॉज ये खुद के सेल को भी, जिसमें वो वर्क कर रहे हैं, उसको भी वो डाइजेस्ट कर देते हैं. नेक्स्ट इज माइटोकॉन्ड्रिया. दीज आर नॉन एज द पावर हाउस ऑफ द सेल because they are the energy creating units, we can say. Mitochondria have two membrane coverings, means they are double membrane bound organelles. Outer membrane is porous while the inner membrane is deeply folded. और वो जो foldings होती है ना, जो outer membrane है that is porous, but जो inner membrane होती है वो simple नहीं होती है, वो फोल्ड्स होते हैं. इनर मेम्ब्रेन फोल्ड्स की फॉर्म में होता है और उन फोल्डिंग्स को हम क्रिस्टे कहते हैं. ये सरफेस एरिया इंक्रीज करता है. These folds increase the surface area for the ATP generating chemical reactions. In generally ये जो inner membrane होती है क्रिस्टे, इसके surface पे ऑक्सीजोम्स प्रेजेंट होते हैं. ये ATP जनरेटिंग यूनिट्स होती है, ऑक्सीजोम्स होते हैं. तो ये सरफेस एरिया इंक्रीज करता है. जितना ज़्यादा सरफेस एरिया होगा उतनी ज़्यादा ऑक्सीजोम्स होंगे, और जितने ज़्यादा ऑक्सीजोम्स होंगे उतनी ज़्यादा हम ATP जनरेट कर पाएंगे. So the energy required for the various chemical activities needed for life is released from the mitochondria in the form of ATP, and its full form is adenosine triphosphate. ATP is known as energy currency because body को metabolic activities perform करने के लिए जितनी भी energy की requirement होती है वो इससे मिलती है. The body uses energy stored in the ATP for making new chemical compounds and for mechanical work. Mitochondria are very strange organelles because they have their own DNA and ribosomes. माइटोकॉन्ड्रिया के जो ऑर्गेनल है उसके स्ट्रक्चर में उसके खुद का DNA भी होता है और राइबोसोम्स भी होता है. एंड इट मीन्स अगर इनके पास राइबोसोम्स हैं तो दे आर ऑल्सो द साइट्स फॉर द प्रोटीन फॉर्मेशन. And DNA है, therefore they are also self-replicating. And माइटोकॉन्ड्रिया के पास खुद का DNA है और राइबोसोम्स है, इसलिए इसको माइटोकॉन्ड्रिया को हम सेमी ऑटोनोमस ऑर्गेनल भी कहते हैं. सेल्फ रेप्लिकेटिंग ऑर्गेनल है. Now the next is plastids. Plastids are present only in plant cells. They are not present in animal cells. So there are generally two types of plastids. Chromoplast, which are the colored plastid, and leucoplast, which are white or colorless plastids. Chromoplast में, for example, there comes the chloroplast. Chloroplast comes under chromoplast. So chromoplast containing the pigment chlorophyll. If chromoplast contains the pigment chlorophyll, they are called as chloroplast, and they are very much important for the photosynthesis process. Chloroplast also contains some other pigment like yellow, orange. The next comes leucoplast. Leucoplasts are generally of three types. Amyloplast stores starch. The second is eleuroplast, which store proteins, and the third is ileoplast, which store oils and fats. So leucoplasts are primarily organelles in which materials such as starch, oil and protein granules are stored. The internal organization of the chloroplast consists of numerous membrane layers embedded in a material called the stroma. These are similar to mitochondria in external structure, and like the mitochondria, plastids also have their own DNA and ribosomes. Plastids also have its own DNA and ribosomes, so it is also a chloroplast also have its own DNA and ribosomes, right? Because it is the plastid, right? So it is also a it is also the semi-autonomous organelle. Also, chloroplast structure is similar to the mitochondria, not actually, but few things are similar. First is that it is also double membrane bound organelle, and the next is that it has its own DNA, its own ribosomes. Now, what is stroma? There is a term here, stroma. So actually, it is a matrix inside the chloroplast organelle, and inside that matrix there are layers which are stacked on each other. The single layer is called as thylakoid. I am talking about the chloroplast structure. The single layer is called as thylakoid, and thylakoids are stacked over each other, and all together it is called, the stack is called grana. So it's the basic structure of the chloroplast. Now the next is vacuoles. Vacuole are storage sacs for solid or liquid content. Vacuoles are small sized in animal cells, while in plant cells there are large vacuoles. तो वैक्यूल्स जनरली प्लांट सेल्स और एनिमल सेल्स दोनों में मिलते हैं, बट प्लांट सेल्स में इनका साइज़ बड़ा होता है और एनिमल सेल्स में इनका साइज़ छोटा होता है. तो एनिमल सेल्स में इनका साइज़ छोटा होता है तो ये इनका नंबर भी ज़्यादा होता है, और प्लांट सेल्स में इनका साइज़ बड़ा होता है तो इनका नंबर भी कम होता है. जनरली इन प्लांट सेल्स द सेंट्रल वैक्यूल इज प्रेजेंट, मीन्स प्लांट सेल में जो वैक्यूल है वो सेंटर पोजीशन एक्वायर करता है और अराउंड वो 50 to 90% ऑफ द स्पेस सेल का ऑक्यूपाई कर लेता है. In plant cells, vacuole are full of cell sap, provide turgidity and rigidity to the cell. Also, जो vacuole होता है, vacuole की जो membrane होती है, vacuole is a membrane-bound organelle, single membrane-bound organelle. So, membrane of vacuole is actually called tonoplast. It is actually called tonoplast. Many substances of importance in life of the plant cell are stored in vacuole. Vacuole एक storage organelle की तरह भी काम करता है, उसमें बहुत सारी molecules store होते हैं और ज़रूरत पड़ने पर vacuole से वो molecules अपने जहाँ पे उनकी requirement होती है वहाँ पे वो transport किए जाते हैं. The molecules which are generally stored is amino acids, sugar, various organic acids and some proteins. In single-celled organisms like amoeba, the food vacuole contains the food items that the amoeba has consumed. Amoeba में food vacuole देखने को मिलता है. Food vacuole जो food particles जो amoeba engulf करता है through endocytosis process, वो उस food vacuole में consume किए जाते हैं. In some unicellular organisms, कुछ specialized vacuoles भी मिलते हैं which play a role in expelling excess water, means which plays a role in osmoregulation and some expelling some excess of the water and some waste from the cell. So हमने अब इतनी सारी organelles के बारे में पढ़ा, and so now each cell thus acquire its structure and its ability to function because of the organization of its membrane and organelle. कोई भी सेल है उसका हमने देखा कि एक ऑर्गेनिज्म का स्ट्रक्चर इतना कॉम्प्लेक्स होता है और इतने सारे फंक्शंस को, कितने मेटाबॉलिक एक्टिविटीज़ उस ऑर्गेनिजम्स में अकर होती है ताकि वो ऑर्गेनिज्म अच्छे से फंक्शन कर सके, अच्छे से वर्क कर सके. तो उसके लिए एक सेल का ऑर्गेनाइजेशन बहुत इंपॉर्टेंट होता है कि उसकी मेम्ब्रेन है, जैसे हम देखा सेल मेम्ब्रेन, सेल वॉल, साइटोप्लाज्म और सेल ऑर्गेनल्स, ये अपने अपने स्पेसिफिक फंक्शंस परफॉर्म करते हैं जिसकी वजह से वो ऑर्गेनिज्म फुली वर्क कर पाता है, फुली फंक्शनल होता है. एंड द सेल इज अ बेसिक स्ट्रक्चरल organization. This helps the cells to perform functions like respiration, obtaining nutrition, clearing out the waste material, forming the new protein. और ये सारे functions के लिए अलग अलग organelles एक cell में present है. Thus, cell is a fundamental structure unit of living organisms. And it is also the basic functional unit of life. Now, the last topic is cell division. New cells are formed in organisms in order to grow, to replace the old, dead and injured cells and to form the gametes required for the reproduction. अब organisms की body में नए cells बनते रहते हैं, either for the growth of the organism or to replace the damaged cells or for the reproduction process. And the process by which these new cells are made is called as cell division. There are two types of cell division. First is mitosis and second is meiosis. Now, first let's talk about mitosis. The process of cell division by which most of the cells divide is the mitosis process. In this process, each cell, which is called mother cell, so this is the mother cell. This mother cell divides to form two identical daughter cells. These are two daughter cells which are identical to the parent cell. The daughters have same number of chromosome in this case. These two daughters have same number of chromosome as that of the parent cell, and therefore it is also called as equational division. This mitosis is also called as equational division because there are equal number of chromosomes in the parent cell as well as in the daughter cells. This mitosis process helps organisms in growth and in repairing the tissues. Now the next is, let's talk about meiosis. Specific cells of reproductive organs or tissues in animals and plants divide to form the gametes. कुछ सेल्स रिप्रोडक्शन के पर्पस से ऑर्गेनिजम्स में डिवाइड होते हैं, और वो जो मेथड यूज़ करते हैं डिवीजन के लिए, दैट इज़ कॉल्ड एज मियोसिस. इस केस में 2 कॉन्जिक्यूटिव डिवीजन्स होती है. मीन्स सबसे पहले मियोसिस फर्स्ट होगी एंड देन मियोसिस सेकेंड होगी. And when a cell divides by meiosis, it produces 4 new cells. ये जैसे माइटोसिस के केस में तो 2 डॉटर सेल्स बनते थे, बट मियोसिस के केस में 4 डॉटर सेल्स बनेंगे. एंड द क्रोमोसोम नंबर इन द डॉटर सेल विल बी हाफ देन दैट ऑफ द पेरेंट सेल. फॉर एग्जाम्पल, ये मदर सेल है, राइट? इस मदर सेल में सपोज 2n क्रोमोसोम्स हैं, तो इन डॉटर सेल्स में ईच वन ऑफ देम विल हैव n number of chromosomes, means half of the parent cell, and that's why meiosis is also called as reductional division. And now with this topic we came to the end of the chapter. If you found this video helpful, please hit the like button, leave a comment below and share it with your friends. And don't forget to subscribe for more such videos. Thank you.